Hello, hello, hello. Oh, hello to you all. Hello, welcome back, and welcome to the True Biology Fan stream, where I, as a true biology fan, will rate every protein. How are all of you doing today? To start off, I'm going to play a little bit of music, but then I'm going to get into some announcements before we get into the rating. And let me know if the music is too quiet. Sounds good on my end, but please let me know if it's too quiet on your end. Hello, hello. RA Dragons, 666th Moab. Music was from this. Did you like it? I'm a big fan of my loading music. So, today, I wanted to celebrate with you all because we hit 1K subs, which was crazy. A lot of people had found the rabies video, which was very fun. And yeah, overnight we hit the 1K subscriber mark, meaning that in a future stream I will be eating a SCOBY, the flat disc of yeast and bacteria that you find in a bottle of kombucha, which is fun for me, but very worth hitting that milestone. Yursi Pest, eating a Whataburger and watching this to procrastinate for my taxes, aka a good night. I'm glad that I can be of assistance with that. RA Dragons, I would make fun of ribosomes, but I don't want Naegleria fowleri to infect me. Yeah, for sure. A maximum AYAB neutron indeed. Not only do we have the 1K subscriber special coming up in an indefinite time in the future, I also have an announcement. And if you've been paying attention to my community posts and my social media posts, you'll know that coming up, I have a collab stream with the psychologist VTuber Sidere. That is going to take place at 10 a.m. EST. It's the channel's first collab, and we're going to be talking about neurobiology, which is going to be very fun. It's sort of in between Sidere and mine's specialty. She is a human psychologist, and I am a molecular biologist that has worked with mice. So it's going to be really interesting getting to experience this paper together, kind of in the middle of our academic specialties. So stay tuned for that. That's going to be quite fun. Joshua, welcome. 1 a.m. stream. Is it 1 a.m. on your end? Thank you for staying up, if this is what you're choosing to do with your early morning. Rabies is basically a real-life horror movie. Yes. I think that there is so to be honest, chat, right? To be honest, I was conflicted with making that rabies video because I don't want my channel to be a place where people go to read or learn about diseases and then feel scared. So I wanted to cover rabies in a way that was not appealing to your sense of fear about viruses and bacteria, but rather one of genuine interest. So I'm glad that even though I chose not to do the clickbaity thing of describing exactly how horrible it was, that enough people showed up, or people seem to have liked the video enough without me having to go like super ham on describing all the horrible things that rabies can do, and keeping it in the realm of immunology, which I hope people appreciated. I have rabies anxiety. Funny about that, when I was a kid and I had just learned about religion, I mean, if you watch the rabies video, I think you know this about me. The fact that the first thing I learned when I learned about prayer was to pray to not encounter any of these horrible diseases. But hopefully, knowing that there is a rabies vaccine out there and knowing that it is, if you can get access to that rabies vaccine, this is essentially a non-issue for you. All right. With that in mind, now that I've done the plug for the collab stream coming up, why don't we get into rating some proteins? Nope, that's hiding me. There we go. Why don't we get into rating some proteins? So before I get into talking about proteins, I want to hear from all of you what your background level, if you're comfortable with sharing, with biology actually is, just so that I know how to tailor my language when it comes to talking about proteins. And in the meantime, I'll catch up on chat. I'm more afraid of prions, though. There's no cure or vaccine, and you can get one anytime. That is true, and this is why I'm so careful with when I'm going to be making a prion video. Because I don't want to make content that makes people afraid. I want to make content that makes people feel hopeful. And I don't want to tease too much about why there's something to be hopeful about in the prion story. But here's some food for thought. Prions accumulate exponentially in the environment. We know that because prions cause other prions to misfold and they survive a lot in the environment. But it begs the question, life has been around for so long, it's kind of interesting to note that we're all still fine. That if prion expansion is exponential, there might be a reason why we're not all already dead. And I'm going to be looking into some reasons why that might be. So even though prions are terrible, horrible, very scary, and I've gotten a lot of comments recently that prions are on the top of their fear list, and I think that's valid, but I think that there is more to be discovered with regards to how prions exist that might give us all potentially a little bit of hope. And I also want to go into research as to how scientists are thinking about how to make prions a non-issue in the future. So stay tuned. If you want to watch a video about prions to feel scared, that's not going to be my channel. I want to make you feel like the human race is incredible in what we are capable of doing. Ebola, let's see that in the list. Yeah, Ebola was actually one of the first suggestions I had on this channel, and I'm definitely going to talk about that at some point. Never heard of prions before. Maybe that's for the best for now. Okay, so let's get into rating some proteins, shall we? Okay, and so the way that I'll do this is down here we have the text, and I will change this text to reflect the category of protein. So the first category that we're going to be talking about are cell cycle proteins, okay? And so what better cell cycle protein to start off with than the proteins of the cyclin class, right? So the cell cycle is, of course, cyclical, right? And so it's interesting to know how a cell can regulate its own cycle just with the use of molecules, right? So, cells are not programmed with code. They're programmed by DNA. DNA codes for mRNA, which then codes for protein. So all of the function within the cell has to work through molecules interacting with molecules, right? And so cyclins are a class of protein that control this circularity of the cell cycles. So the process by which cells divide and create more cells. And this is an extremely complex, coordinated event. And if it goes wrong, you can end up with things like cancer. So it's really, really important that everything in the cell cycle happens exactly the way it needs to happen. Which is why cell cycle, which is our regulation of the cell cycle, is so important, right? And so cyclins are a group of proteins that bind to enzymes called cyclin-dependent kinases. These are enzymes that chemically modify proteins to go through the cell cycle. The cyclins themselves also go through cycles. So not only do the cyclins regulate cycles, they are also themselves regulated in cycles. Cycles. I'm going crazy with that word. But the idea is that during the cell cycle, the level of these cyclin proteins will increase or decrease, and their specific concentration will regulate exactly what happens during that cell cycle. If you're really into the aesthetic beauty of interconnected systems, then I think that you would find cyclins to be amazing, right? If you love thinking about how things interact on a kind of conceptual level or like a numerical level, then cyclins are definitely going to be up your alley. But because I get to inhabit the space of a biology enjoyer and not your teacher, I can actually have an opinion. So instead of being like all biology fields are equally valid, I can actually just say that I much prefer thinking about molecule-to-molecule interactions. And so the cyclical nature of increasing levels and how that feeds into like nodes and pathways and regulation is kind of not my thing. So I'm going to go ahead and put this into not my taste, I'll say. So let me go ahead, change the size. Probably have to go smaller than that. Whoa, too big. Okay. Cyclins will go into not my taste. Okay. Cholera or influenza. So I don't have a particular opinion on what I would cover first, although I did do a little bit of reading into cholera, just batting around ideas for what to cover next. For those of you who... I'll save discussion of cholera for when I eventually talk about cholera, I think. Do they regulate their own cycles or do they regulate each other? That's a great question, Joshua. I'm unprepared to talk about cyclins. I've written a couple of sentences for each one of these proteins, but you can imagine that nobody actually has all of biology loaded up at the same time. And in fact, that's a really good segment into me talking about the skill set required for being a biologist, right? A lot of people assume correctly that memory is really good for being a biologist, and that's certainly true, but actually memory is not necessary. It's not necessary for success in biology, which might surprise people because there are so many things in biology, but what's more important than your ability to memorize is sometimes your ability to teach yourself things, right? So a little known secret is that not all of us have all the information loaded up at the same time, but if you gave me like 30 minutes, I could probably figure out that answer for you. But yeah, if I learned more about cyclins, maybe I would put them higher, but from my memory, from what I understand about cyclins, it's just not my thing. I prefer visually thinking about how molecules interact with one another rather than interconnected systems. But I acknowledge that it is interesting for a lot of people. Obviously, biology is always interesting for the biologist that studies it, right? Toxo, I vote flu. I'm biased towards viruses over bacteria. I actually was the same way, but I'm beginning to be much more of a bacteria appreciator over time. The reason why is that they are a lot more, I don't know, sympathetic. That seems kind of weird as a as a neutrophil that I would feel sympathy for bacteria, but you might learn why when I release this gonorrhea video, which I'm still working on. So, cell cycle, that was actually the only cell cycle protein that I had on this list. These next couple are going to be cytoskeleton proteins. Hi, Riley. Welcome to the stream. We just started. If you've seen Cyderay's social media, you'll know that there's a collab coming up tomorrow at 10 a.m. where we're going to be talking about neurobiology. But now we're transitioning into cytoskeleton proteins, and this is a category that I have had viewers write in their own proteins to be put in this section because there are some pretty hype cytoskeleton proteins. And I believe the first on the list that we're going to tackle is alphabetically earlier than the rest of them. It's going to be actin. Okay? Actin. Okay, actin is a part of the cytoskeleton. This is a picture of actin that has been stained with different fluorescent markers. And so typically, a good marker for actin in the cell is phalloidin. And phalloidin is a compound. It's a toxin that actually binds very tightly to actin, and you're able to make it glow red, basically, under a microscope. And so this is what you're seeing are actual threads of this cytoskeleton protein in a cell, which is, I just love looking at actin. It's always very aesthetic. Just for that alone, for having such a beautiful cell staining pattern, it deserves to be not in the bottom of the tier, I'll say. So, what does actin do? Actin forms microfilaments, which are essential components of the cell's cytoskeleton. And it's largely responsible for things like muscle contraction, cell movement, and a lot of things. As a motile cell myself, as a neutrophil, as a cell that gets to move, actin is incredibly important for what I do for a living. And so the way that actin functions is through dynamic growth and disassembly. So for those of you who have watched my shorts, you'll know that actin assembles, so actin monomers, so the individual components that make up these threads assemble and disassemble. They assemble on one end and fall out of the other. So you get this treadmilling effect. So as these threads are building, you'll get pieces of actin that stick on one end, and from the other end, those pieces will fall off. And if the rate that actin is assembled is faster than the rate it is disassembled, threads grow, and vice versa, right? If actin monomers disassemble faster than they can grow, then overall that thread will disappear. But if you get the concentrations just right, you can get a filament that appears to move because you get assembly on one end and disassembly on the other end at the exact same rate, so it looks like the filaments are moving. Kind of like frost walking in Minecraft, which I found to be very, very interesting. This phenomenon is called treadmilling, and for that alone, I have to put actin in the interested tier. I'm definitely not obsessed with actin, but I acknowledge that it has aesthetic beauty, and the mechanism of treadmilling is one that I find to be very fun, so I'll put it in the interested category. Ursipest. So what you're saying is neutrophils and tanks move the same way. Caterpillar tracks, actin tracks. That's not quite what I mean. By treadmilling, you can imagine, so unfortunately, I have no idea what's like free use and what isn't on the internet these days in terms of getting to show you. But if you look up actin treadmilling, you'll see what I mean. So it's less like a literal treadmill and more like if you had a stack of balls in a line, removed one from the end, and then added it to the other end. You would see that if you continually did that, then that filament would appear to move. So the way that cells move is that you get a lot more assembly on one end of actin, and that assembly kind of pushes the membrane of the cell forward, so it kind of allows us to stretch our membranes. And you can kind of see that in my design because I have all these fluffs around me. I'll go ahead and make myself a little bigger, kind of representing the way that a neutrophil would move through the body are these protrusions that actin filaments push against to deform the membrane, which I think is super cool. Let me resize myself. Yeah. So I definitely like actin. Wow, I have legs, yeah. Don't often get to see these puppies, do you? Yeah, so I definitely like actin. It's not my favorite of the cytoskeleton proteins, but it's definitely worth mentioning just for the the growth and disassembly. Very neat, very neat. Tubulin. Tubulin is next. Oh, that's not right. Tubulin. Tubulin, alrighty. Give me a second. Okay, so tubulin is another cytoskeletal component. It's a major component of the railways of the cell, called the microtubules, and it's important for mitosis as well. So, you can basically think of tubulin as making up the cell's public transit system. So, you know, there are a lot of people that would find that itself to be quite interesting, but for me, that's not quite enough, for me personally, right? But I still rank tubulin kind of high, because tubulin has a unique molecular property called dynamic instability, which, I mean, I don't remember who said it in the last stream, but biology has some of the sickest names in science. Dynamic instability. And the thing is that's not even an example of scientists making the name cool on purpose. Dynamic instability is actually very descriptive, right? It's dynamic in the sense that you can get growth and disassembly very quickly, but unlike actin, which assembles on one end and disassembles on the other, you get these railways that build up like tall towers and then fall apart from the top to the bottom, creating dynamic instability. You build up these railways, and then once you finished building that tube, there can be an event where the entire tube collapses. So this is another thing that you should absolutely look up because it looks really cool. Dynamic instability, peeling away these tubes that build up the cytoskeleton. It goes kind of underappreciated, I think, in undergrad biology, how dynamic, changing, and uniquely alive the cytoskeleton actually is. So you should look that up. And when the microtubules fall apart, that's called catastrophe, which again, I think is just awesome. Suggestion, if it hasn't been added yet, complement protein, specifically membrane attack complex C9. Yeah, I'm already ahead of you. I have a whole section about immunology. Hard to visualize. Yeah, I want to be really careful about making sure I have permissions to show everything. So unfortunately, for some of these, you're going to have to use the power of imagination. But just know that the components that make up the backbone of your cells are remarkably dynamic, and they themselves feel remarkably alive. Okay, so tubulin is going to be ranked very similarly to actin, where I'm not like obsessed with tubulin, but I do acknowledge that it's very cool. Okay. Then next on the list is intermediate filaments. So you typically learn about all of these cytoskeletal components kind of in the same breath. Here's the thing about intermediate filaments, is I don't remember a whole lot about them. I do remember that they are very important in cell-cell adhesion. So they make up a good proportion of the molecules that bind cells together, which can be quite important if you're thinking about cancer, because you don't want cancer cells to breach certain barriers, metastasize, and travel the rest of the body, right? So they're interesting for that regard. They also make up a lot of the biology that you can see plainly with your eyes, right? So this is a major component of keratin. It's a part of your hair and a part of your nails. But by definition, almost, they are, I mean, you can tell, right? The name is already so vague, right? They are quite varied, and it gives me, it makes it hard for me to determine, you know, what they are conceptually as a group because they're so diverse. So I'm gonna have to put intermediate filaments in the don't know enough category. I mean, I did go through grad school. I did at some point learn a lot about them, but for some reason or another, they just haven't stuck with me quite as much as some of these other proteins on the list have. Okay, now we get into some proteins that I find to be very, very interesting. And the first is going to be this protein, dynein. Okay, so dynein is a motor protein. And it's a motor protein that I feel gets less love than its sibling, I guess you could call it. That's going to be the next protein that we review. So some of you biology fanatics will already know the protein that I'm referring to. I'm of course referring to kinesin. But dynein is really cool too. So what do I mean by motor proteins? Motor proteins are proteins that walk on top of the microtubule railway. So this isn't really like an exaggeration or something that is like alliterative, like a what alliterative, that's not the word. It's not like a simile, metaphor, or fancy imagination thing. These proteins literally walk, they put one foot ahead of another across the microtubule railways, and they carry cargo on their backs. So they are very much like alive in their own regard, right? They move, and they move like insanely similar to how a human would walk, right? They're bipedal. I also find the structure of dynein just to be kind of neat. Like you see in, I guess what you would call like the chicken thigh of the dynein, you have these beautiful structures, these, I guess this would be like a hexagonal kind of thigh thing that seems very interesting. And so dynein, I don't think deserves to be anywhere below kinesin in terms of how much I think about dynein. In fact, if you've watched the rabies video, you would actually know that dynein is a part of rabies' story. So dynein actually gets hijacked by the rabies virus to go travel up the neuron. So it's co-opted by pathogens just as much as kinesin is. Joshua, the railways being the tubulin from earlier, correct. Isn't this the thing that rabies uses? Correct. Rabies riding the dynein down the brain, up to the brain. Yep. All that is true. So I hope that dynein gets some more love amongst biology students or people that have taken biology, because kinesin is the motor protein that takes all of the love, right? Kinesin is so popular. And why is that? Well, if you are a biology student, right? If you are a biology student, then you will have seen, if your teachers had access to the means to show you, you will have seen The Life of the Cell, which is an animation that Harvard Medical School put out. And this is an incredible 3D animation from like a decade ago that still holds up very, very well. And in that video, in The Life of the Cell, they have an extraordinarily well-modeled animation of kinesin walking down a microtubule, carrying a big vesicle on its back. And it's beautiful. In fact, I would say that that video alone is maybe more responsible for how many people choose molecular biology than we would like to admit. Like, it made me very excited when I was in high school learning about kinesin for the first time. In fact, I have to put this in the obsessed tier because I actually, in undergrad, joined a lab that studied this protein, so at some point I knew a lot about this. And so I'm very happy to put kinesin into the obsessed tier, right next to dynein, its cousin, sibling, whatever you want to call it. Just, to me, you can tell that my taste is, if I can imagine or see a molecule move in interesting ways, that's going to be what determines what I think is interesting. Yursi Pest, what's up? What can I clarify for you? J. Andre, I failed biology, but I'm here anyway, so hi. Very happy to have you. There are many ways, I think. So for those of you who don't know, I am like, in my life, a biology teacher. And so I think a lot about, how to best help students through biology. And I think that there's part of it that is just biology is a lot of memorization, and that's really, really tough. And, some good questions in chat before I go on this tangent. Joshua, how is it different from dynein? That's a great question. So dynein is I believe it's a minus end motor. So it always travels in the minus direction of tubulin. So tubulin has a plus end and a minus end. That's just to distinguish the fact that tubulin has endedness, right? So it's not identical at both poles. So it kind of has like a north and south pole, right? So dynein only travels towards, I guess you could call the south pole. So it generally travels, maybe I shouldn't mix metaphors too badly. It generally travels towards the cell body, I'll say that. And kinesin generally travels away from the cell body, I believe. Somebody who might be more on top of that in chat might correct me later, but I believe that's the endedness. Basically, they travel in different directions, is what I'll say. I work in high school, and the biology teachers here completely skip over the cytoskeleton entirely. That's such a shame, because the cytoskeleton is, especially dynein and kinesin, like if you want students to be interested in biology, then you have to show them the narrative potential of biology. That's my personal opinion. A lot of people disagree because there are a lot of educators, especially at the higher end, higher education end, who believe that you should never assign narrative to science. And I think that's reasonable, right? Like, it's very tempting to believe that evolution has a goal, right? It's even useful to think of evolution as having a goal, right? But if you believe that evolution has a goal beyond getting species to survive, then you end up with people who think that evolution always makes things better, because that's what narratively makes sense, right? As a story, in Pokemon, generally, for 90%, 99% of Pokemon, when they evolve, they become stronger, better, faster. But that's not what evolution is. Evolution is more about fit. It's survival of the fittest, not survival of the fittest in the sense of, how fit you are when you go to the gym. It's survival of, if the world is a square peg, are you able to fit in a square peg? Is your biology compatible with your environment? And if it's not, then there are mechanisms by which species evolve to better fit that environment. And so there's a risk that people take away the wrong message, because if you think that evolution always makes things better, faster, or stronger, then you might end up thinking things like, actually, you know what? I don't actually want to get into that right now. Maybe for a different stream. The idea is, if you think that evolution always makes things stronger, you are wrong. And so there's a danger to assigning a story to biology. And I acknowledge that, right? When you get to a very high level of understanding, you have to remove the narrative, the urge to assign narrative to science, in order to get it right. But at a lower end, I shouldn't call it lower, at an earlier stage of your biology learning career, I think it's fine, because it generates interest, it generates passion. It's helpful to hear stories that you like. It makes learning easier when you like it. On today's episode of I don't know anything about this, but I'm super interested, evolutionary biology. Yeah. Evolutionary biology, for me as a molecular biologist, serves more as like the intro, right? So it's important to have a grasp on evolution, just because evolution is at the core of all things biology, and understanding that will help you a lot in appreciating the storied history of biology. I went off on a huge tangent, but basically, dynein and kinesin move in incredibly lifelike ways, and I think it's a shame that your high school doesn't cover the cytoskeleton, because when I've talked to students before, especially the ones that are interested in biology, they all know that video about kinesin, and you can see that their faces light up, because it evokes something, childlike glee, I think, to watch something walk that you don't expect walking. Okay, moving on. A lot of proteins to get through. Okay, myosin. Okay. Okay, let's get myosin on the screen. Myosin. Okay. Myosin is an actin-based motility protein. It is a motor protein, but instead of being a motor protein for tubulin like dynein and kinesin are, this motor protein acts on actin fibers. And so this is the protein that is responsible for muscle contraction. So basically, a 101 level explanation of how this works is that there are like layers of actin filaments that make up your muscles. And in between these layers, you have these myosin proteins. And these myosin proteins have a very simple job. All they do is a power stroke. So basically you see these heads. Go ahead and move my model over it. You see these heads. And these heads can actually retract. So they can kind of, like if you take your palm, your hand, and you lay it flat, if you curl your fingers in towards your palm, that's the same motion that myosin is capable of. So if you have these motor proteins that are in between these sheets of actin, this motion of the power stroke can actually cause those actin filaments to slide over one another, causing them to be shorter, which decreases the length of the muscle, which allows that muscle to contract. So this is mechanically at the core of how your muscles work, which is interesting. But for me, it's not quite as evocative of childlike wonder and glee than dynein and kinesin. So I actually, unfortunately, have to put myosin in the not that interested category. I think I also have bad memories of this particular protein because one of the reasons why I did not become a medical doctor is because I found physiology to be quite difficult. And in that class, we did cover, muscle structure and how muscles work. And I did not do that well in physiology, which might surprise you, because of how deeply I love biology. But maybe, that's a part of the grudge that I have against myosin. Do I know anything about the etymology of affixes attached to these proteins? Does that mean the origin of the names? I actually don't know. For some of them, there will be interesting stories. Like for cyclin, I actually forgot to tell this story, but cyclin was named by the scientist. So basically, the scientist that named cyclin really liked cycling, like the sport, like riding a bike. And so they called it cyclin because of the obsession with cycling. It was actually a coincidence that cyclin also happens to be cyclically regulated, which I found pretty interesting. Myo means muscle. Yeah. Thank you, Toxo. Yeah, that makes sense. Great. Yeah, okay, so I think that's actually it with these cytoskeleton proteins. Speaking of evolution, how evolutionary conserved are some of these proteins? Did kinesin emerge in mammals, animals, eukaryotic, all life? That's actually a great question that I don't really have a great answer to. If I had to guess, I believe all of these so far that I've mentioned are highly conserved, at least across eukarya. So, for those of you who don't know broadly the domains of life, you have prokaryotic life, which includes things like extremophile archaea that like live in hot springs and bacteria. And then there are eukarya, which are pretty much everything else. Multicellular life. So, I should know this. But I don't want to do any kind of misinformation. So again, the trick of being a biology teacher is that, the day before you teach your lesson, you cram all of the information and then appear to be an expert the following day. There are like 30 proteins on this list, so that wasn't really possible for me to do. But it is a really interesting question, right? Like, these things appear to be very important for the basic function of the cell, and they certainly are. But whether or not, for example, a bacteria needs myosin is actually a very interesting question, but not one that I am actually prepared to answer at this very moment. I love it when I actually get questions that I can't get answers to, not just like on stream, but in class too, because when you get a question you can't answer in class, it means that the student has had an idea that surpasses your knowledge or memory at that time, which means they've pushed against what is supposed to be a more advanced person, which I think is always very exciting. If a student has an attitude about getting one over me, that annoys me. But I'm generally very happy to be like, I don't know. Like, that's a great question. You've reached the boundary of my knowledge. And I think that more teachers should do that. Never really thought of the capacity that proteins have for actual kinetic motion. Yeah. You should definitely look up the videos for kinesin and dynein movement. And if you watch my rabies video, then you've already seen the dynein movement. Thank you to Medbunk for allowing me to use that video, which is very nice. Hello, yes, who just writes nerd. Thank you, I wear that with a badge of honor. You love the experience of getting a question you can't answer in the history department. That's awesome. I'll go out on a limb and ask if your username is referring to the Black Plague and if that is the area of history that you're most interested in. Hello, Thunder Nugget. Hi-fi. Glad you can make it even this late. Thank you for showing up. Yeah, we're rating proteins. We just got finished with cytoskeletal proteins. Basically, my summary, if you're just tuning in for the cytoskeleton portion, is that the skeleton of your cells is like alive on its own. Things walk, things move, things build, and things disassemble. If you like watching molecules move, the cytoskeleton is awesome for that. But we're done with the cytoskeletal category. We are now moving into development. Okay, developmental biology is a field of biology that I have a complicated relationship with. Developmental proteins. Do you have some sick-ass music to learn proteins from my muscle gain? Well, I'm playing Lo-Fi's playlist right now, but if you want a recommendation, you can watch my tardigrade video. The first 30 seconds of music was made by my friend. I asked her to make a funk track that I could use, and that's what she came up with, and it's a banger. Okay, so we'll talk about developmental proteins. Okay, so developmental biology, let me just preface this, is that I have a very bad memory. My short-term memory is actually just like 5 units. So you can measure your short-term memory online with these tests that basically get you to repeat a few digits back after briefly seeing them, right? So if someone were to give me a phone number, it would take me a long time to memorize it, because the most I can do for repeating back a phone number is 5 digits, which is really small, which is why I say, like, you don't have to have like an amazing capacity for memory to be a biologist, but you have to like, so I just have to work harder than everybody else is basically what I'm saying. In the my colleagues all have great memories. They can memorize things very quickly. It takes me a while. But developmental biology is a field where memory is so important because it's so interconnected and variable. And I struggled a lot learning developmental biology, not only in undergrad, but also in grad school. But the reason why I say my relationship is complicated with developmental biology is because I find it to be so awesome. I think developmental biology, while I was learning it, was amazing. I just hated being tested on it. Yuki, hello. Hello, Entropic Alloy. Thank you for showing up. Antibodies, yes, antibodies are on the list. I'm doing it category by category, so I don't have to reteach basic things about the fields in general, but antibodies are definitely on the list. don't worry, we will get to them actually pretty soon after I finish the developmental section. Okay. So first, let's get into Hox proteins. So, if you've watched my shorts, then you would know that Hox proteins and the Hox genes are largely responsible for coordinating your body plan. So why does your arm grow in the position that it grows? Why does your head appear on one end and your other end appear on the other end? That is controlled by a group of proteins called the Hox proteins. And so the Hox proteins are transcription factors. These are proteins that determine whether or not a gene is turned on or turned off. Oh, I forgot to mention. I've been kind of bad about this, but I'm also going to shout out the viewers that suggested these. So kinesin was actually suggested by a viewer, Eric Martin. So I'll continue to do that as we go down the list. But anyway, Hox proteins, they're transcription factors. They turn genes on or off. And so these regulate and are really important regulators of genes during development. And so these proteins are essential for determining where your limbs develop. And so the picture that I have selected is a picture where if I, unfortunately I already merged these layers, but on this fly, instead of antenna, these flies have grown legs. So you can actually see it kind of over here, that this fly that is developed normally has regular antenna. They're kind of thin. You'll notice that this quote-unquote antenna is way thicker. That's because this fly has a mutation in the Hox genes that told the spot on the Drosophila head to become a leg instead of an antenna. So the fact that you can get this level of control with Hox genes just through one mutation is absolutely nutty, right? That's insane. And once you realize this about biology, that in these organisms, these mutations can cause like a pretty straightforward swap, you begin to appreciate both how simple biology is and how complicated it is. Because we don't know everything there is to know about Hox genes. We're still figuring them out, right? But the fact that one mutation can just swap limbs is like weirdly close to video game logic, right? It's almost as if your model like glitched. And I think that's super, super cool. So I'm actually going to put Hox genes all the way up into the obsessed category. You'll notice that if I've done a video on the topic already, that it probably already is in my obsessed category. But yeah, so Hox proteins are incredible. Very, very interesting. Okay, I'm going to take the opportunity to now catch up on chat. Oh, I guess I'll load up the next protein so that you have something cool to look at. And that's going to be in the notch proteins. Okay. Notch proteins. Now I will catch up on chat. Antibodies, yes, we're doing antibodies. I'm not prepared to talk about individual immunoglobulin. Yeah, I'm not equipped to talk about individual immunoglobulins at this time. Yuki and Thunder Nugget, you both appear to know quite a bit about immunoglobulins, so I'd be happy to hear what you have to say if there's something particularly interesting about them that you would like to share. In Tropic, when I was an undergrad, we had a bunch of PSET questions that involved Hox proteins. Yes, very complicated. I would not be able to explain mechanistically how they work at this particular moment, although I can give you a basic understanding. Antimicrobials I don't really think fit in this particular stream. I might have a different stream entirely that deals with them. Okay. It's a little disturbing. Legs for eyebrows. Yeah. Yep. Hox proteins. Very, very bizarre. How do Hox signal where the arm grows? So, Hox proteins, like a lot of the developmental biology proteins, works on location-based expression and concentration. So if a certain protein is produced in a certain part of the embryo at a certain time, at a certain concentration, that will determine whether or not the correct genetic programs are activated to make a certain body part. And so I would not be able to explain to you bit by bit how exactly that works, but that's the general idea of it, right? So spatially, temporally, concentration-dependent expression of these proteins will affect what grows where. And until you take an advanced level developmental biology class, that might be what you have to be satisfied with for now. Is this Minecraft? Yes, this is Notch Minecraft. This protein was not named after Notch of Minecraft, but I just thought that it was a funny little coincidence. I do have a real picture, but I just wanted to put that up with no explanation. So the Notch proteins. The Notch proteins are cell-cell signaling proteins. So they are membrane proteins that lie on the outside of cells, and they help cells talk to one another. So this is a set of proteins that are highly conserved in animals. So these proteins, as you know, the category implies, are really important for development, and so they also help polarity. So here's an interesting question for you, Chad, and it's an interesting question that I think points to why I find developmental biology so fascinating. Imagine an egg, right? How does an egg know which end of the egg is going to be the head and which end of that egg is going to be the tail? I want to actually hear from chat if you have hypotheses, and if you know the answer, then keep it to yourself. But if you haven't heard of, if you haven't learned about developmental biology, how might you expect polarity to be established? I want to hear your hypotheses. Other proteins have fun names like Nemo involved in the TLR pathway. True. So Yuki, it seems like you're quite interested in immunology. It appears to be. Joshua, liquid going to the bottom of the egg. That's not a bad guess, honestly. There are mechanisms by which polarity are established that do kind of have to do with a conception of gravity. So yeah, it's interesting to hypothesize how polarity is established. And so for some organisms, polarity is established through a polarity induced event, like the sperm entering the egg, right? Like that has the potential to inform the egg which end should be the head and which end should be the tail, which is really interesting. Lectins are cool too. They're like antibodies that can be derived from plants and attached to carbohydrates. So I am woefully underprepared to talk about anything plant related, and I blame that on my education. I'm very trained in animal biology, because most of the choices that I made during my educational career have been towards eventually wanting to be a doctor for humans. So I would love to hear more about lectins at some point. The concentration gradient of a certain protein named after a certain blue hedgehog, a protein that we will be talking about very soon. Is Notch the same as JAK-STAT? I believe they are related, but I honestly don't remember too much about JAK-STATs. Good question though. Okay, so cell-cell communication, it helps establish polarity. Polarity itself is an interesting problem. Maybe barometrically, the wide end of the egg would experience more internal pressure than the thinner end. That's also a really interesting possibility. I don't know if there are organisms that do that, but the reason why I bring up so many interesting possibilities is because, surprisingly to me, the mechanisms by which polarity is established is not that conserved between individual species. The way that a Drosophila egg determines whether or not the one end is the head or one end is the tail is actually really different than how a mouse does it, which is surprising because polarity is something that you would expect is really important and therefore really well conserved, but it's not, and that's really, really crazy. I will go over complement, we'll go over immunology after the developmental section. Okay, so is there anything else about Notch that I have? Yeah, cells touching cells, regulating the symphony that is development. Polarity is interesting. So I'm gonna put Notch in the interested category, but I don't really know enough about it to be obsessed. I do find cell-cell polarity to be a very interesting topic though. Okay. So that's a Notch protein. Here we go. So this is a protein that was teased by Thunder Nugget, and it is, of course, Sonic Hedgehog protein. It amazes me, addressing the elephant in the room, that a shitposting scientist got to name one of the most important proteins to ever exist after this blue hedgehog. And this is especially like SEO relevant because of the new Sonic trailer, which people have been quite hyped about. But Sonic the Hedgehog is so important for regulating the symphony of development. And as Thunder Nugget had said, yes, Sonic Hedgehog is a concentration dependent regulator. So areas of the developmental embryo are informed by their level of Sonic Hedgehog, whether or not they should be one end or the other. In fact, so this is a is the hedgehog part real? Absolutely. Sonic Hedgehog is absolutely the correct name for this protein, although I believe that in a medical context, it's either just referred to as Sonic or Hedgehog. I believe it's actually just Sonic, because if you watched my Weird Genes short, you'll know that, you know, biologists are off here goofing off and having fun, and that's all well and good, but the second that a doctor has to talk to a patient, those goofy and fun gene names are no longer appropriate. So I'll still call it Sonic Hedgehog forever because that's awesome. There's also a protein called robotnikinin. I don't actually know if it's a protein. There's a molecule that is an inhibitor of Sonic Hedgehog. It's called robotnikinin because biology is awesome and we get to name things all sorts of great fun ways. Pikachurin is also a great one, yeah. Another draw for being a biologist is if you find a new protein, you get to name it. Where was I? So here's a little secret. I don't know if I will do this video. It's such a good video idea. I'll keep it a secret. I'll keep it a secret. I'll keep it a secret. What I'll say for now is that one story that I can tell about Sonic Hedgehog is that Sonic Hedgehog is one of the proteins that is responsible for determining how many spinal segments you have, which is really interesting. And it does this in a way where it... How should I illustrate this? So we're all familiar with the concept of ping, right? The idea that, like, you can ping a network and the network pings back at you. Sonic Hedgehog, when determining the number of somites, which are segments that will eventually become your spine, will turn on and off at this, like, cyclical rate. And these waves of Sonic Hedgehog pinging, these like pings of waves that radiate out from one end of the embryo, will inform that embryo how often to split the embryo into somites. And the reason why I'm dragging my feet on that video is that I would love to animate it, but I'm just not good enough at editing quite yet to do that idea justice. But Sonic Hedgehog is super important in developmental biology. For that, I will put Sonic Hedgehog into the obsessed category. You will definitely be hearing more about Sonic the Hedgehog from me. Okay. Actually, Okay, so we have like one protein in between development and immunology, but let's skip ahead to immunology and I'll come back for that other protein. Phi, shouldn't your nucleus be segmented? So, you don't know what my nucleus looks like in the plane that goes into your computer. I could be hiding my lobes behind the screen. Let's go into the immune system now. So we've talked enough about development. Hopefully I've convinced you at this point that developmental biology is like sick. But what's more obviously sick is the immune system. Get it? Sick. Immune system. Haha. Yeah. So we're going to talk about immune system proteins. And these are, of course, crowd pleasers, right? I mean, you cannot... I don't believe that there is a biologist out there that isn't interested in the immune system, because I think, again, it has the most salient narrative. So we're actually going to start with the complement proteins. So let me go and try to find those. Okay. Immunology has the best narrative. It's also really hard to study because it's really freaking complicated. But I've done the crunching of the information for you, so you get to just enjoy the cool parts while I break it down for you, right? So, the complement proteins. The complement proteins blow up microbes. They're the immune system's way to blow microbes up, and that's not an exaggeration. And this is also not something that gets covered in your 101 biology class, right? I never learned about complement protein in high school, and I believe in undergrad we maybe touched upon it. It was only in grad school, in my lab, where I learned about how complement proteins work, and it's such a shame. Yeah, they do more than blow cells up, but as an entry point into understanding how cool they are, this is awesome. So basically, they're a set of proteins that lead to the formation of what's called the membrane attack complex. And again, this is another entry into why biology has the coolest, sickest freaking names ever, right? Membrane attack complex. And this complicated pathway eventually forms these proteins that like binding bacterial cell membranes, opening holes in them. And the thing about biology is that it's really important to keep the inside stuff separate from the outside stuff, because having the outside stuff flood the inside stuff means that the inside stuff is no longer at the correct concentration for things to work the way that they should work. So these complement proteins literally blow holes into invading bacteria. And so, as some commenters have said, these proteins are also responsible for triggering all of these inflammatory pathways. So they're very important in the immune response. So, I'm actually going to put complement proteins up by obsessed. I love the idea of the immune system making these protein complexes that blow up cells. It's so cool. And blowing up cells in a different way than apoptosis, because I know I've talked about apoptosis pretty much ad nauseum on this channel. In various forms, but this is a blowing up of a different variety, so I think that's awesome. Definitely deserves a high spot there. I believe that there was a viewer who recommended that I cover this before the stream, but I don't actually have the name down. Okay. You're supposed. What cell type produces complement proteins? That's a good question. So I'm not actually that prepared to talk about that particular question. Again, 30 proteins, a lot to get through. If I had to guess, B cells are often the cell type that produces a lot. Well, so they're like the antibody proteins, basically. So they're also responsible for mediating other immune reactions, but I don't actually have that information at the top of my head. But it looks like Yuki might be pretty enthusiastic to share the correct answer. Incorrect. Yep, that was a guess. If you would like to put the right answer in chat, I would be very grateful. Okay, so let's next talk about antibodies. This is now something that we are all maybe painfully aware of. So let me load those up. Antibodies. Getting a little crammed up here. Okay. So we're all pretty painfully aware about what antibodies are by now, having endured a certain plague that will not be named. But there's a little more to antibodies in terms of why I find them to be particularly interesting. So these are Y-shaped proteins that bind specifically to certain proteins. In the immune context, this can trigger the tagging of bacteria, the jamming of important proteins, activation of the immune system, and we also extensively use antibodies as a research tool. And so, why I find antibodies to be particularly interesting is I want you all to think of this particular question. How much variability, or I shouldn't say that. And if you know the answer to this question, please don't spoil the answer. I want the non-biologists to sort of ponder this, right? Antibodies. Your cells make antibodies. And your cells have to make antibodies that bind proteins that your body has never seen before. And that's an interesting problem, because we know that the blueprint for our proteins, all the proteins that we are capable of making, the blueprint is written in our nucleus. It's written in our DNA, right? I'll get to that question, Yuki, in just a second about how antibodies neutralize pathogens, but I won't go into too much detail. Again, there are a lot of proteins to get through. But before I get into how antibodies neutralize pathogens, I just want the chat to ponder this fundamental question. How is it possible for your body to create proteins that bind proteins that your body has never seen before? So Yuki has actually spoiled the answer in the chat, VDJ recombination. But thankfully, that is jargon, so we can still think about how that might be possible for a little bit. Just to sit with that question, to be a little uncomfortable, I think, is a part of what makes learning interesting, right? So we've thought about the problem for a little bit. It's a unique problem that the body has to solve. It's actually a problem that a lot of organisms have to solve. Wasn't in that Kyrgyzstan video. I actually don't watch Kyrgyzstan anymore because I don't want their content to influence how I make my content. You're a pest. Random intake from the environment. Isn't that the whole argument that getting little infections is good for your immune system? So those are two separate thoughts that might not necessarily relate to one another. They make all of them and more as they get used, right? Not quite. It is an interesting problem. And it's something that we take for granted because for most of us, antibodies are just something that we make after we get a vaccine, right? I want you to think about a different question. How much variability in your DNA do you think exists inside your body? In other words, do all of your cells have the same DNA? There are notable exceptions, right? The sperm and the egg, once they are fully matured, have half of your total DNA payload. Definitely true. So sperm and eggs are exceptions. Their DNA looks very different than all of the other cells. But what about the rest of your cells? Random muscle cell, random neuron, etc., etc. how much variability do you think exists? Apart from random mutation. For the most part, your body's cells, for the most part, have identical DNA. With a couple of exceptions. Yes, epigenetics aside, yes. Epigenetics aside, all of the same base coding in one organism, they all are the same, except for certain cells in the immune system. Except for certain cells in the immune system. And so this is getting into VDJ recombination, which is something that Yuki brought up, which is how the body learns to fight infection. So the way that this happens is in developing T and B cells, a specific region in their DNA is allowed to scramble itself to form an unimaginable amount of diversity. So in T and B cells, in their DNA code, there is a section that is allowed to be scrambled. And by scrambling that DNA, T and B cells have different DNA than the rest of your body. So they have unique instructions that none of your other cells have to generate copies of antibodies that are unique. And so if those T and B cells encounter and bind a foreign antigen, they will be allowed to mature and make those antibodies. I find that to be incredible. It's an incredibly creative solution to this problem, right? This problem of how does the body learn to identify something that it's never seen before? And the process by which it does it is by scrambling DNA all the time. And if you get a hit, if you get a match, then those cells are allowed to survive and those cells are allowed to pump out antibodies for you. So, hopefully, if you are able to, you've gotten your COVID vaccine. What that vaccine will do for you is it will generate a piece of the COVID virus that your immune system can identify. The immune system then shows that protein to the T and B cells. And the T and B cells that have, by chance, scrambled their own DNA to make the correct antibody will survive and then help you make those antibodies for the rest of however long you're able to maintain that immunity. Fascinating. I think that strategy by evolution, when I think about it, that to me is the definition of an idea that I am obsessed with. Just insane, right? The variability that the VDJ recombination allots you is incredibly variable. 10 to the 11 different antibodies can be generated, but it's not infinite. Which is maybe why some vaccines are more or less effective than others. It is mind-boggling, 10 to the 11 as a number, but it's not infinite, which is interesting as well. Do lymphocytes scrambling some DNA and gametes scrambling all of their DNA use the same protein mechanism to do it? So I believe those are different mechanisms. So in terms of your immune cells doing it, they have this machinery that allows them to, and specific regions that allow them to do this. Gametes would be the process of meiosis, which would be different machinery. Mm-hmm. Why not infinite? So the region that the antibody is coded by a combination of different segments, and those segments can only be recombined so many different ways. So it's not actually infinite. I don't actually have an immunoglobulin. I'm not actually an immunologist. I'm a general molecular biologist at this point, but I would love to do a deeper dive on immunoglobulins in the future. What gene? I did not write the name of those genes down, unfortunately. There are different segments that I don't remember which segments those are. Yeah, so the purpose of this stream is to give people an overview of all the proteins. I'm not prepared to engage at an expert level on each individual entry that I have in here. But for giving me a way to talk about VDJ combination and the fascinating way that the body deals with foreign antigens, I'm going to put antibodies in the obsessed tier. Oh yeah, thank you for reminding me. So we know that antibodies generally help us fight back infection, but there are certain ways that antibodies can do that. And again, I'm not going to be going too deeply into that. One way that antibodies can help the immune system fight back is to tag bacteria. So to give them a little tag that immune cells can kind of stick onto and react to. But also something called like opsonization. So you can get blocking of important proteins that the bacteria might need, or blocking of important proteins on a virus that the virus might need, and that like shutting off of, or that separation of the pathogen from the rest of the organism is a part of the story of how the immune system fights back. Don't immune cells make temporary plasmids for signaling during recombination? I'm not entirely sure. I don't think so, but I would have to look more deeply into that. Okay, this next protein I am so excited to talk to you about. I spent quite a bit of time studying this particular protein. It is MHC, otherwise known as Major Histocompatibility Complex 1. So, no, it's no problem that you wanted more greater detail. In fact, I want to go into greater detail, and I will, in fact, for a lot of these stories. But like, think of this as the introduction to a very long story. It's just not possible for me to hold that much data in my brain at the same time. Joshua, you're gonna leave. It's 2:30 a.m. where you're at, and you're falling asleep. Thank you for coming. Thank you for spending your time here learning about biology. It really means a lot that there are people willing to stay up that late just to chill and talk about stuff that I really enjoy. I hope you have a good night. Major histocompatibility 1 is a core component to the adaptive immune system. So you have the innate immune system, which, you know, as a neutrophil, I am a part of. So I would be the front line, the innate immune system, the immune system that counters everything broadly. But this protein MHC1 is core to the adaptive immune system, the immune system that contains, I guess you could call like the snipers of the immune system, the specialists, the one that go in and precisely neutralize targets, right? MHC is a very important protein for them to do their job. So what does MHC do? And I'm specifically going to talk about MHC1. MHC displays antigens. It displays stuff that has been digested inside of the cell. So you can think of your body, and it's as if, like, every time you ate a pizza, that a patch of your skin would form a pepperoni. Something like that. It gives the immune system a way to sense whether or not a cell is infected or not. So if a cell has, for example, eaten a virus, then the cell's digestive system would chew that virus up, and MHC is responsible for showing the immune system, hey, I recently have been infected by a virus. I'm showing you a piece of that virus. I need help. So then the T cell, which are cells that basically force cells to explode. Explode is maybe a gratuitous word, but T cells force cells to commit apoptosis. So that the cell that is infected is no longer a fertile breeding ground for whatever threat is replicating inside of it, right? There are so many little mysteries about this protein and so many interesting immunological stories that have occurred around this tool. So I want you to think about this evolutionary story, right? And I'm going to simplify this because, of course, it has to be simplified. Otherwise, we would be here all day. But think about the evolutionary arms race, right? So imagine a gazillion years ago where the adaptive immune system is in its early stages of development. And you've got a problem. And the problem's name is viruses, right? These viruses think that they can take up the free real estate in your body, and your body doesn't like that. Even if the virus itself isn't malicious, because again, you can't really assign agency or narrative to a lot of these things. The virus wants to do what the virus wants to do, which is to replicate and pass on its DNA, right? But the issue with that is that by being infected with a virus, the virus uses your stuff. It takes up space. It co-opts the cellular machinery. You have an evolutionary incentive to rid your body of this virus. So what do? Over evolutionary time, a system evolves to recognize whether or not a cell is being taken advantage of by a virus. And that system is, of course, the adaptive immune system. So the T cell comes in, and the T cell evolves to recognize this protein MHC, which can either display its own guts, so the regular gut flora, you could call it, of the cell, or it can display a viral protein, and then that triggers the immune system to react. So you can think of this as a back and forth war. So that is that is one step of the evolutionary story, right? So at this stage, we have the upper hand, right? We evolved a protein that teaches the immune system whether or not a cell is infected, and now the immune system has a way to specifically snipe out those cells that have been infected. So, what do you think viruses evolved? I want to ask this question to chat. Hypothesize, if you were a virus, what you could do to really mess this system up. And it can be as vague as you would like, right? Just think for a sec. If you were a virus and you have this powerful tool that you have to sabotage, how would you sabotage it? You're supposed to stop the cell from producing MHC at all. Exactly right. So there are viruses that have evolved many different mechanisms to reduce the level of MHC. And how might it do it? There are some viruses that produce proteins that pull the protein out of the membrane, that literally rip the protein from its place and directs it to the cell's digestive system. There are viruses that stop the genetic programming that demands the production of MHC. There are viruses that prevent MHC from traveling to the membrane at all. Along MHC's journey, viruses have adapted so many different ways from allowing that protein to make it to that membrane. So what do you think the immune system evolved to do? What do you think the clever immune system did to combat viruses that reduced the level of MHC on the cell surface? I'll give you a second while I hydrate. We're an hour and a half talking about biology. I hope you all have the time to stretch and to hydrate. But it's an interesting question, right? They produce MHC themselves for cell uptake. So basically, your hypothesis is that the cell maybe works overtime to make MHC2. I actually don't know if that counts as a correct question, to be honest, Thunder Nugget. I'm not prepared to say whether or not that's correct or not. No, the clever immune cells. The clever immune cells developed NK killer cells, natural killer cells. And these cells evolved to detect MHC. So if these NK cells bump into a cell and they don't have MHC, they blow the cell up. Isn't that fun? That's crazy. This back and forth is so exciting. When the NK cell detects MHC, it stops it from blowing the cell up. So if you don't have MHC on the cell surface, that destructive programming is not going to be turned off, and then the NK cell will blow up the quote-unquote infected. But I'm not done yet. Okay, I'm not done yet. What do you think viruses evolved to do to work around NK cells? So the back and forth is, we make this MHC protein, it sticks viral peptides on the cell surface, T cells blow up infected cells, viruses lose. Viruses evolve a way to pull MHC out of the membrane, viruses win. We evolve NK cells to blow up cells without MHC, viruses lose. What do you think some clever viruses evolved next? Take another sip of my tea. Some viruses evolved a way to make their own MHC, or to make their own version of viral peptides that look and mimic your natural your natural flora. So they hide by saying, no, you know, this cell's perfectly healthy. It's got MHC on the surface. Meanwhile, it's the virus that directed that MHC there and loaded it with a peptide that would not trigger the immune system. I would love to make a whole video just on this story on itself. But I could not share that bit with you live because I feel like, you know, being forced to sit with that uncomfortable information of that unknown, I think, is part of what makes getting the answer so much more interesting. So obviously MHC is gonna go right up into the obsessed category. I love that protein so much. Okay, I can catch up on chat now. Are MHC genes the same as VDJ? I don't believe they are. I believe MHC genes are like multi-allelic, meaning that there are all sorts of different combinations of specific versions of MHC that you can inherit, but I'd have to double check that. So it's not exactly that it is like a scrambling of the DNA, but rather that that particular gene has so many variations. Yuki, I'm not actually prepared to expand upon exactly what that MHC story is, but again, these are like teasers for future videos, right? I would love to make just a video on that, and I hope I got that story right. I'm pretty sure I did, but I will correct myself in a MHC only video if that's the case. Okay, so that is actually the last immune system protein. Let's quickly, for just one protein, head back into DNA. I guess I can call it, yeah, DNA replication. This will be a short one. So, this protein. Get everything back into order. Topoisomerase. So, I probably should have put the polymerases up here. I forgot to, and there was a viewer who wanted RNA polymerase up here, but I'm not like ready to talk about that. Polymerases are cool. I think I'll probably do a couple of protein tier lists. This is not going to be the only one. Obviously, there are like a zillion proteins. These are just ones that I wanted to talk about this time. So briefly, what I wanted to say about topoisomerases is that there is a class of protein whose job it is to unwind DNA. And the reason why I decided it would be fun to mention this here is just to get a glimpse for the kind of audience that I have. You know, if you are someone who is obsessed with topology, like if you're a math kind of person, I bet you would find topoisomerases quite interesting. Because, you know, DNA has this complicated structure, this double helix that's wound up. And in order to make copies of that DNA, you need to unwind that DNA and separate the strands, right? And so, one of those classes of proteins that helps you to do that are the topoisomerases, which nick the DNA in a certain spot and allow that unwinding to actually begin. So it reduces the kind of tight strain on that system. Yeah, I'll probably talk about DNA polymerase on a different tier list. But here's the thing, I don't really care. I put that up there in case anybody that likes topology is in chat. It's not my personal thing to get interested in. So I'm not really interested, and I don't actually know enough to move it higher than that tier. Okay. Topology is cool, but it makes my brain hurt. Yep. Topology, fun, but if I think about it too much, it just hurts. Okay. So let's get in now to the next category, which is going to be metabolism. So this time I only actually have 2 metabolism proteins on the list. Let's do metabolism. These are also going to be, yeah, actually this is not short. What about ligase? Again, there are so many proteins. I'm not going to get to all of them today, but ligase would be a fun one to do in the future. So let's do ADH. So, alcohol dehydrogenase. So, I think for fun, I put this in here just so that I could rant a little bit about genetic variability. So, in humans, this enzyme converts ethanol, which is drinking alcohol, into acetaldehyde for continued processing in the liver. So it's basically what stops you from like poisoning yourself, because ethanol, when you drink ethanol, ethanol is poison. Don't let all the studies about red wine fool you. Ethanol is definitely just poison. And so alcohol dehydrogenase, ADH, is an enzyme that allows you to partially detoxify that ethanol. So what I find interesting about this, and why biology students in general have this enzyme in mind, is that this enzyme gives professors a way to talk about competition, competitive inhibition. And what does that mean? There's this idea that there are enzymes, and those enzymes' functions can be multifaceted, right? Because in biology is dictated by things having form and function, right? But form and function isn't necessarily very specific all the time. So enzymes are very specifically shaped, but they aren't generally perfect in terms of the things that they break down. So, there's a very memorable episode of House where House is treating a patient that voluntarily consumed, I believe it was wood alcohol, aka methanol. And methanol is very poisonous. Methanol actually gets converted into formaldehyde. Not formaldehyde, but I think formalin, which is a very, 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 very toxic chemical. And so this patient voluntarily consumed this wood alcohol to, we'll say, prematurely shuffle off of this mortal coil, right? And so House is treating this patient, and yes, Yusupest, you've got it. House takes shots of ethanol, drinking alcohol, with this patient as this patient lay dying. Because ethanol and methanol... Oh, he drank printer ink. Sorry, I got that wrong. Okay, sorry, I misread your message. Yeah, in that episode, yes, drank printer ink because it contained methanol. But the solution was the same. The solution was taking shots of ethanol. And the reason why this works is because the space that alcohol dehydrogenase has for these alcohols is flexible enough to fit both ethanol and methanol. But if you drink enough ethanol, the drinking alcohol that is less poisonous for you, that'll kick the methanol out of that protein and prevent it from being converted to formalin, saving your life. Now, nowadays, you don't have to do that. There are other ways to prevent methanol poisoning. But that story always sticks with me because it's cool. It's just cool. So even though I think that, I don't know if that's really a merit of the particular protein in general. Like, I don't know if it deserves to be in a high place just for the fact that it was cool in an episode of House. So I'm controversially gonna put this down as not interested, besides the like competitive inhibition thing, because that's more of like a medical discovery than a biological one. Is methanol race car fuel? I don't know what methanol is used commercially for. We use it in the lab sometimes, but I don't know what the average consumer would know methanol, in what context. Okay, so that's my little spiel about ADH. Cool episode of House. As an enzyme, the function for me is just kind of whatever. Okay. Next metabolism protein, and this is another one that I think is a banger. It's ATP synthase. Okay. So I am like omega not ready to talk about exactly how ATP synthase works, because just knowing how this enzyme works is like a whole chapter in biochemistry. And again, with 30 proteins, didn't have the time to relearn all of biochemistry, right? But there are certain things I learned about ATP synthase that make it worthwhile to talk about. So, how does the body generate energy? You need to create ATP. ATP is cellular fuel. There are chemical properties that ATP has, this molecule ATP, that make it very useful for powering the enzymes that give you life. And so this enzyme has to be at least one of the constructs that are responsible for making that energy. But it's kind of like getting job experience, right? How do you get a job without job experience nowadays? And this is kind of the same thing. How do you make fuel from an energetic standpoint, right? It's kind of an interesting problem. Because anything that would require you to move a protein in a certain way is going to require energy itself. So how do you get a system where you generate more energy than you put in? And that's the problem that metabolism solves. I'll just call it metabolism in general. And I know that there are viewers that wanted me to put the complexes of the citric acid cycle on here. I'm not like super ready to talk about those. Maybe in a different tier list I can bring this up again. But these things in general coordinate to basically generate this energy for you at a net profit. And the way that they do this is by taking advantage of electrochemical gradients. So instead of burning ATP to make ATP, which is not, an efficient use of your cellular fuel, the citric acid cycle forces the cell to create a sort of concentration imbalance. And so because all systems want to return to homeostasis, the resolution of the separation of gradient powers this enzyme. So this enzyme isn't powered by ATP, which a lot of enzymes are. This enzyme is actually powered by hydrogen ions flowing through the protein. So things in general flow from a high concentration to a low concentration, and this constant flowing of hydrogen atoms down this enzyme is like pulling a ripcord through a Beyblade. I think that's like the best way for I don't know, for my boomers in chat who remember what a Beyblade is. And that's incredible, right? So it doesn't actually need the same fuel that the rest of your enzymes need. It can generate its own energy through this ripcord effect by the resolution of the gradient. And it is an extremely beautiful enzyme structure. Ripping this ripping the protons through this enzyme actually causes this enzyme to rotate. So it actually really is a lot like a Beyblade. And the internal structure as this enzyme rotates squeezes ADP with another phosphate, creating ATP. So the proton pump, when that gradient is resolved and flows back through ATP synthase, it generates the ripcord that you need to move the enzyme in a way that generates ATP for you. And to me, that's a very, very beautiful way of solving this problem of how do you make ATP without burning ATP. And of course, the honor doesn't just go to ATP synthase, it goes to the entire citric acid cycle, but I didn't put on the citric acid proteins. Maybe next time. But ATP synthase, for solving the clever problem of cellular energy, goes into the obsessed category. And I actually don't have that much more space in the obsessed category, which is maybe going to be problematic as we get down to the tier list. I might have to shuffle things around and remove things. Okay. I'm gonna catch up on chat a little bit. Isn't ADH a hormone? I think that there are annoyingly multiple different uses of that abbreviation. CC Mustang, thank you for joining us. What do you think about the fusion protein, BCP-ABL1? I actually don't know what that is, so if you can inform me what that actually is, I might be able to offer an opinion, but given the fact that I don't know what that is, chances are not looking good. Yeah, the structure of ATP synthase is crazy, especially from the top-down view. Highly recommend you Google that if you are able to. Okay. Here is the section that everybody and their mothers are waiting for, and that is neurobiology. Okay? I know for a fact that this next protein is going to be something that people are really interested in, and it pains me to say that I will keep most of this research to myself because I do want to properly explain this in the full video. And many of you can probably guess it's the prion protein. Okay? You know it, you hate to see it. Arguably terrifying proof that maybe God doesn't exist, or at least has a terrifying sense of humor. This protein is commonly known for its terrifying effect when it is misfolded. So something that people forget about proteins, especially the prion protein, is that your body makes this protein. And it wouldn't do that if it wasn't evolutionary advantageous for some reason. So people forget this protein actually has like a real function in the cell. But we more commonly know this as the responsible agent for a whole bunch of prion diseases, like mad cow disease, like chronic wasting disease. Prion proteins, when they are misfolded, cause other prion proteins that are otherwise normally folded to misfold. And so you get a accelerating aggregation of these proteins. And what is terrifying about this process is that these prion proteins rearrange themselves to form tight beta sheet structures. And these tight beta sheet structures have so much surface area and they're packed so tightly that the body has a really tough time getting rid of them. And that's why these prion diseases are essentially a death sentence. Because heat, because enzymatic degradation, none of those are effective when it comes to ridding the body of this garbage that builds up. And so, if you are to take this mechanism to its logical conclusion, then you would realize that this would eventually mean that there will be an exponential growth of these proteins accumulating in the environment, because nature doesn't seem to do a great job with destroying them. But it leaves us with a question of why we're still here today, because, you know, exponential equations grow very quickly. So it's still possible that we're in kind of a latent period for the prion disaster, that maybe we are just lucky to be alive exactly when we are. But in the prion video, I'm actually going to propose something that might calm you down, a potential alternative explanation. And I don't want to spoil that here, but just so that you aren't racked with anxiety over how terrifying these proteins are, there might be some reason to believe that we'll be able to deal with them. Of course, you know, through science. But enough about prion diseases. Let's talk about what they might do in the nervous system. We don't really know, is the short end of it. It's really hard to study this protein. We think that it is involved in sleep, which might explain why fatal familial insomnia is a result of a prion disease. We think it might be important for memory, and it might be important for neuron outgrowth, meaning the growth of the protrusions in the neurons. And that's a little concerning. It's a little interesting. I just think in the field of prion research, the fact that we still don't really know or we don't really have a clear idea as to why we're making this protein to begin with, which I think is very interesting. As someone who is like more into biochemistry, on the biochemistry side, I'm going to put prion protein in the obsessed tier. And don't worry, I'll have more interesting things to say about prion proteins in the future. But right now, I think that I just don't want to give too much of that away. And now I'm finding that I'm going to have to reshuffle some of these. So, unfortunately, I'm going to have to demote Dynein just because I know more about kinesin. Should I? This is so hard. Okay, maybe we keep Dynein up here. These are all so good, is the thing. Okay, ATP synthase, I'll put down into interested. I'll keep prion protein up here. Yeah, okay. I'm thinking I'm happy with that. Can we fold prions? Thank you, Renee, for joining. Can we fold prions? So, I'm going to interpret that question as... So there are things called like molecular tweezers, which allow you to suspend molecules. I don't necessarily know that we as scientists can like physically fold them, but we can try to add chemicals that will force proteins to fold one way or another. Another set of proteins that I didn't have up here are the chaperone proteins. These are proteins specifically evolved to help proteins fold correctly. So once these prions form these aggregates, once they misfold, they really don't like being folded back into the correct orientation. If you Google the prion structure before and after folding, I don't think they have a full crystal for misfolded prions, but you can tell you go from a lot of alpha helices, which have this kind of shape, if you follow my cursor, and a majority of that protein then becomes this zigzag pattern. And that's a pretty dramatic change in terms of how a chaperone potentially could refold that protein. Separating dynein and kinesin is blasphemy. Sorry. Oncogenic fusion protein. I see in our lab all the time. Also, we see notch a lot. It's cool how two different proteins can merge into something completely new. Will I cover defensins/interferons? Not on this stream. There are a gazillion proteins. I will keep in mind all these other suggestions. I think we'll have plenty to talk about in another one of these protein tier lists, if these are streams that you all appear to be enjoying. What else did I have to say? In terms of fusion proteins, I want to be very careful here, but I believe fusion proteins are completely a man-made construct. So it's something that scientists have artificially programmed to combine two different proteins as a research tool generally. I'm not entirely sure what the function of that oncogenic fusion protein you're talking about actually is, so I would not be able to answer that sufficiently. Okay. I think I'm gonna skip tau, because I actually don't have a lot to say about it at this moment. But I do have other neuroscience-y stuff that you all might be interested in. Let's pull them up. Here we go. Okay. AMPA/NMDAR. These are maybe not the most commonly known proteins, but these are essential for any neuroscientist to be familiar with. So, we know that neuroscience is going to be that we are on the cusp, or in a neuroscience revolution where our molecular understanding of what our conscious experience is improving. But in order to reach that level of understanding of like what makes the mind, we all have to learn about these two receptors, because fundamentally, at the core of understanding how neurons work is understanding not only how they fire, but how they quote-unquote learn, or how they change their firing pattern. And by learning about AMPA and NMDA receptors, it's in your like journey of understanding the brain, one of the first times you get to molecularly understand what it means for neurons to change, which is very exciting because it starts to demystify a lot of what we might assume about the brain. So, generally, I think we can accept the fact that, you know, from an experiential point of view, if you don't use it, you lose it, right? And anecdotally, this is true. If you know several languages, if you don't speak that language in a while, you will like lose the ability to speak that language. And neurons work kind of similarly, where if you have correlated firing, meaning one neuron fires to another neuron, and then that neuron also fires, that's correlated firing. If you have that happen often enough, then the strength of the signaling between those two neurons will increase. So basically, one neuron is going to have an easier time getting that other neuron to fire. And that's the basis of how neurons strengthen their connections. On the other hand, if two neurons don't fire in a correlated manner, in other words, if one neuron fires and the other one neuron doesn't, you start to lose that connection. So neurons that fire together, wire together, neurons that don't fire together, don't wire together. This is not the same as learning and forgetting, but the analogy is helpful because actually decreasing the strength of neurons is really important for memory as well. So it's not that straightforward as forgetting and memorizing. But these two receptors, there's a lot to get into with how they work. But what I'll say for now is that these receptors, or at least the NMDAR receptor, is sensitive to the amount of ions that are in and out of the cell. So the NMDA receptor is a major regulator of neuron strengthening and neuron weakening because it can sense the amount of activity through sensitivity to ions. So it's kind of electrically sensitive, you could say, in a way. And once there is a certain threshold of charge, a cation, I think it's a magnesium ion that blocks the NMDA channel regularly will be kicked off of the channel. And that's going to allow calcium to influx into the cell. And once that calcium enters that cell, calcium is going to trigger the genetic programming that creates more receptors on the surface of the neuron. I don't have time to get too deep into how that happens, but if you can understand the general idea that the more firing happens, the more electrically different that environment becomes, something in that triggers the NMDA receptor to then allow calcium to inflow and then change a whole bunch of genetic programming in the neuron to make the neuron more sensitive or less sensitive to firing. Is there a neurological reason why neurons don't have gap junctions? Ooh, that's a tough question, because that is... So here is my hypothesis. This is speculation, okay? For those of you who don't know, gap junctions are ways that cells can communicate with one another, but there's a caveat. Those cells have to be attached. They have to be right up next to each other, physically touching, for those gap junctions to allow for direct cell-cell communication. It's a great question, but I love this question. My hypothesis as to why this didn't evolve for neurons is because the brain is made up of more cells than just neurons. There are supporting cells like microglia, like oligodendrocytes, like astrocytes that provide support for the neurons in many different ways. And I believe that these supportive cells require access to the synapse, the gap between neurons, in order to do their job properly. I believe they also play a role in regulating neuronal function, and if they didn't have access to the synapse, if they couldn't also sense the neuronal activity, they might not be able to do their various jobs. I believe that's the explanation, but I'm not 100% sure. Great question. Okay, so I really like AMPA and NMDR receptors. I think the mechanism is kind of interesting, so I'll just put it in interested. I don't think it goes up into obsessed for me. Okay. Oh boy, how many more of these do we have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Oh boy, 12. Okay. There are some of these where I don't have as much to say, so hopefully some of these I'll get through quite quickly. Unfortunately, that's not going to be this next one. So this next protein is another neuroscience favorite. It's amyloid beta. So this is what we believe is the causative agent responsible for Alzheimer's. Like prion protein, it is a protein that aggregates, not easy to get rid of. Junk collection in the brain isn't particularly great. And so when this junk builds up in the brain, you start to get neuronal death. And so, like the prion protein, it's not exactly obvious like why we have this protein. In fact, mouse knockouts, if you knock this protein out, doesn't really seem to be that big of an issue for them. But we think it might be important for synapse repair, because the amyloid beta protein is a membrane protein. What I find interesting, and I want to do a whole video about amyloid beta and Alzheimer's in general, but I'll give you a little teaser here, is how incredibly difficult it is to study Alzheimer's, because Alzheimer's is a disease of age. And to me, that's an interesting problem because most animals don't live that long. if you didn't know, mice don't get Alzheimer's. Their amyloid beta just doesn't misfold. Is it because they don't live long enough to misfold? I don't actually know the answer to that. But a lot of our research in mice has to be done in these very genetically modified mice that express a lot of copies of human amyloid beta, of fold susceptible amyloid beta. And there's a big question in Alzheimer's research as to whether or not it's even worth using these mice, because A, mice don't get Alzheimer's, and B, we pumped them full of our human version of this protein. So whether or not that has validity in real humans is a serious question that the field has contended with. Amyloid beta on its own, I don't think is actually that interesting. I think the disease is more interesting. So just like topoisomerase, I'm going to put amyloid beta down here. It's possible that I don't know enough about it, but actually I don't think anybody knows really enough about it to really understand. Okay. Sodium channels. How are we hanging in there, guys, by the way, for those of you who are watching? Still hanging in there? We're 2/3 of the way done. Sodium channels. Okay. This gives me the opportunity to talk about charge and electricity in a biological context, which I think is interesting. And I'm going to start off this conversation in a way that is maybe seemingly not related to our discussion here, but for those of you who are watching, how do you think an electric eel generates current? How do you think an electric eel makes electricity? How does something biological generate charge? If you're like me and don't have a strong understanding of physics, then you probably have a question mark going above your head, right? Ooh, I actually have a thing for this. Yes, you're probably like me doing this, right? I'm not a physicist, so the mechanisms by which electricity work are not entirely clear to me. But it's interesting to note that electric eels that can generate electricity do so with specialized cells that have evolved from neuron and muscle, because we forget that we are actually generating electricity all of the time. And so when you study neuroscience, you learn that the neuron is an electricity generating kind of cell. It's just that the eel takes it up to 10. Can you explain how streptavidin and biotin have such high affinity together? Not off the top of my head, unfortunately. But the voltage-gated sodium channel is maybe the first time in canonical biological education, where you begin to view cells as having electric potential, which I think is very interesting. If you are interested in the brain, this is one of the first things that you learn, and the mechanisms by which it works are quite well studied. It has these beautiful different sharp phases, and it is at the fundamental mechanistic heart of how neurons function, how they fire towards one another. The thing is that I think it's fine, in terms of whether or not I find it interesting. I find it really important, but it doesn't really tickle my brain like the rest of this other stuff does. So we put it in not my taste here. Okay. So this one that is coming up, we are now officially done with the neuroscience category. We're going to move on to the other category, and Thunder Nugget, you are here at a pretty good time, because this next protein is actually one that you suggested, and that is luciferase. Let me go ahead and pull that one up. Okay. Let me make sure I'm on the right layer. Okay, take this down. Okay. Luciferase. So, luciferase, from my perspective, is primarily used as a research tool, although organisms use it to generate light. There are some cool things about this enzyme for sure. So, it's named after Lucifer, whose name means the light bearer, which is already pretty sick. That's already pretty badass, right? Luciferases are actually, and I didn't know this before until I looked this up, but they're actually very diverse. So we give the generic name luciferase to enzymes that can break down the substrate of the enzyme to generate light. So different organisms may have had different enzymatic strategies to accomplish that. So actually luciferases don't have like a conserved generalized form. But the way that they generally work is that they'll break off a piece of their substrate, which is luciferin, to produce light. This reaction is actually, and this is very surprising to me, apparently 80 to 90% efficient, whereas a light bulb is about 10% efficient, and my source for that is Wikipedia. I forgot to mention, a lot of what I'm talking about, again, you can't have all of this information in your head at once. Wikipedia is an excellent resource to refresh on. So the fact that luciferase is wildly more efficient than what we can engineer right now, I think that's really, really cool. In terms of where I would place it, I'm sorry, Thunder Nugget, but I'm gonna have to put this in not my taste. I acknowledge that there are cool things about luciferase, but in terms of the things that appeal to me in terms of my own taste, it's it doesn't like keep me up at night, so I'm gonna have to put luciferase in that category. And I would love to talk about taste, by the way, in terms of how to develop scientific taste or how to discover your own scientific taste. It's something I'm thinking about doing in the future. Okay. Hello, Jerry. Hello, Jerry. Welcome, welcome. GFP. So because you mentioned luciferase, I figured that it would also be good to talk a little bit about GFP. So GFP is one of biology's most used molecular research tools, and this is a protein that was, I believe, isolated from jellyfish. And so this protein, if you can't tell, has just a beautiful barrel structure. And at the center of this barrel is what's called I believe it's called a fluorophore. It's a molecule that allows fluorescence. So if you shine a light, the correct light, onto GFP will give off a different light than you shine on it, so that makes it fluorescent. And I put it here because I want to acknowledge its like beauty in terms of its structure and its use in biological applications, but beyond like fluorescence, I'm not like a chemist or a physicist, so I don't find like the process of excitation and emission to be that interesting. So I'm going to put, I guess I do find it interesting that biological molecules can produce this kind of effect, but it's just, for me personally, my tastes, I don't believe that there's much there that tickles my fancy. The real question is, what does luciferase taste like? So if I ever hit 1 million subscribers, I will do a stream where I taste various biology reagents and buffers. That is something that I made clear for my debut stream. But I'm still confused. What's up, Toxo? Where is the confusion? GFP, the print statement of microbiology. True, true. The spiraling beta sheets do look super aesthetic. Yeah, I mean, a lot of proteins are just blobs, but GFP looks designed, like it looks pretty nice. Plus a ton of other fluorescent proteins derived from it. That's true. That's true, Jetty. There are a lot of research tools that were made from GFP just by swapping out that fluorophore in the middle, like RFP, which is red fluorescent protein. Oh, I'm still confused. You're right. Sorry. I'm so bad at piloting the expressions. Thank you so much, Shoxo. These are the questions I would wonder about in micro. Yeah. I mentioned this on a different stream, but the broth that we grow bacteria in kind of smells like ramen if you open your mind. Of course, you're not supposed to sample it, but I will at 1 million. We are on the way there. We did hit our 1K subscriber mark, which was very exciting. Okay, next up on the list. Okay, hemoglobin, I'm going to talk about real quick because there's not much to say. Okay, hemoglobin, it's got this beautiful structure where it's made out of 4 subunits, so it's a tetramer. And each one of these subunits can bind oxygen. And so every biology student who has taken biochemistry will perhaps cringe at the mention of hemoglobin because this is the thing that is used to illustrate this concept of cooperative binding. So, the way that certain proteins and enzymes work is if one thing binds, it then makes the other things easier to bind. So if one oxygen binds hemoglobin, it makes the next oxygen easier to bind, making the third oxygen easier to bind, and then the fourth oxygen easier to bind. What makes hemoglobin interesting is that it's this cooperativity difference between maternal hemoglobin and fetal hemoglobin that allows the mother to feed the fetus oxygen, as far as I remember. It's a pretty neat fact. In terms of studying Michaelis-Menten rate reactions, I found it to be one of the less interesting parts of biochemistry for me in particular, so I'm going to put hemoglobin into the not interested pile. I know a friend's lab mate that drank yeast extract as a protein shake. That's actually not that hard of a flex because you can buy yeast extract for food at the grocery store. So actually interesting, if you didn't know, if you're trying to go vegan, then yeast extract is an excellent way to mimic Parmesan, according to vegans. But it is very savory, kind of like Parmesan. Hemoglobin is boring. Literally any other blood protein has more going on. I'm actually not too familiar with blood proteins in general, so by default, hemoglobin is the best one for me right now. But if you want to offer a suggestion, I can add it to the growing list of other proteins to look at. Okay, so next here, we have a user write-in from a user just called B, which is bilirubin, which this is a molecule that I had no knowledge of. So I had to learn about this de novo. Let's see. Although I'm not entirely sure if I have it on the list. I am sorry, B. I will put it on the list for the next tier list that we do, because I don't believe that I ended up putting it on the list. Can't find it. Sorry. Never mind. We will not be doing bilirubin. Proteasome. Okay, the proteasome. Technically not one protein, and More Juice is the user that suggested this, so you actually came just in time. Another very aesthetic protein. Asterisk. It's not one protein, but a protein complex. And oh boy, Photoshop is chugging right now with how many objects are, or how many layers are on the screen. Should I merge everything? That doesn't actually seem like a good idea. Maybe we'll just try to make it through. So, More Juice, you can correct me if I get anything wrong, but the proteasome is a protein complex, and I like to think of the proteasome as the cell's wood chipper. It is a protein complex that detects proteins that need destroying, and then chews them up in like a wood chipper fashion. Like this is like a barrel of death that proteins go to die, basically, which I think is pretty metal. I think structurally what an interesting question is why doesn't the proteasome destroy itself? I didn't have the time to really look into that answer, but I do really, really like the proteasome. It also wraps into the ubiquitin story, which we'll get into in just a little bit. So actually we are now in the protein processing category. Whoa, typing. Protein processing. So I really like the proteasome. I gotta say that I don't know enough about it to put it in the obsessed category, so I'll just leave it in interested for now. Maybe one day when I decide to do a video on the proteasome, I will teach myself all the goodies about that. So that one was more juice. Okay, the next one is also a viewer suggested protein, Kuro Gude Kuma. So this is also a VTuber, Kuro. Let's get that up there. Don't think I put that in there too. We'll just do some text for that. Because I do want to talk about this real quick. Ribosomal proteins. So, the ribosome is made of a combination of RNA and proteins. And what I find interesting about ribosome... so before we get into that, the ribosome is the complex that is responsible for stitching together proteins. And the catalytic part of the ribosome is RNA. And so, if you watched my RNA stream, you'll know why I find this so fascinating. The fact that a molecule whose primary purpose is to propagate information also has some ability to catalyze chemical reactions is incredible. I neglected to mention in that stream that the ribosome is not just RNA, but it's also made out of protein. So to me, it's actually really interesting that evolution has kind of converged these two classes of proteins to form one molecule, because most proteins are just made out of amino acids and maybe some complexing ions and some other small molecules. But ribosomes are kind of special in the sense that they are complexes that merge RNA and protein, and I find that very fascinating. So I'll say it's interesting, but it borders on like not my taste, because I couldn't go further than that. Or actually, you know, I think this would appropriately be don't know enough. I think that's more accurate in terms of my relationship to how much I know about those. Okay, ubiquitin. I'm excited to talk about this. I already talked about this on my last stream, but ubiquitin is a part of the protein processing pipeline that allows the proteasome to do its job. So there are enzymes that stick this little tag called ubiquitin onto proteins that need to be destroyed. And part of why I want to mention this is if you were not in attendance at my last stream, this is going to blow your mind, is that Araki, famed author of the very popular JoJo's Bizarre Adventure, had the opportunity to design the cover of Cell. What is Cell? Cell is a journal, a biology journal that is one of the top journals in existence. If you're a biologist, you want to publish in Cell. It's highly prestigious. And Araki got to draw the cover for one of the issues. You can just search Araki Cell Journal. And Araki's depiction of tagging ubiquitin onto proteins that need destroying is fantastic. It's amazing. It's mind-blowing that Araki got to do that. Ubiquitin itself, though, I'm gonna have to put in not that interested. I think that the entire like story around ubiquitin is interesting, like protein processing as a whole, but ubiquitin itself, gonna have to put in not that interested. Okay, we are actually almost done, if you can believe it. There are two more. Okay, snare proteins and phospholipid scramblase. And I'm sorry to Riley, I don't know if Riley's still in chat, but Riley is the one that recommended this scramblase, and it happens to be the last one on the list. But first we'll do snare proteins. Okay. Snare proteins. Oh boy, okay, I'm like running out of breath. Let's get this on a top layer so that we can see them better. So there's a very interesting problem that the cell has to solve in order to deliver proteins from one end of the cell to the another, or between cells, right? So if you can imagine, here's what you have to know in terms of why I find the snare proteins to be interesting. There's a chemical, physical problem with membranes. And if there's one thing that you might remember from biology, is the word phospholipid bilayer. And you might even remember that one end of the membrane, so we'll just, point here. Oh God, why is that doing that? Oh no, that's just on my OBS, that's fine. You might remember that in membranes, that one end of the lipid is positively charged and one end is negatively charged. So if you want to deliver cargo, if you want to send out little packets of membranes, the cell has to solve the problem where this side of the vesicle that contains the cargo that you want to merge with your cell, this side of that membrane has the same charge as the membrane you're trying to fuse with, right? And that's an interesting problem, right? Because if you just jam them together, if you just push them together, these two things, the vesicle that contains the cargo and the cell which you want to deliver to, if you just bump them together, they're going to bounce because they have they have the same charge, right? So positive attracts negative, negative attracts positive, but positive repels positive, negative repels negative. So there's a very serious problem with how... There's a very serious problem with a cell in terms of merging vesicles. And the snare protein. Snare proteins, this is a class of proteins, help overcome that massive energetic barrier. It's very, very chemically difficult. Very, very chemically difficult to make this merge happen because of their chemical properties. But snare proteins allow that merge to happen. And, I've been saying, I enjoy biology that I can visualize. You can imagine that the snare proteins are like threads that wrap into each other and tighten. So you get this mess of snare proteins, and they'll actually rearrange themselves in ways that forces the vesicles to fuse. Is snare an acronym for something? I don't remember. It definitely is. Snap receptor, apparently, according to Nobody Important. Interesting. Yeah, so you can imagine physically this happening, and you can watch videos of this on YouTube, of these proteins actually flipping and tightening, physically dragging these vesicles together, which I think is a very interesting mechanical solution to an energetic problem. I really, really liked learning about these, but it doesn't reach the same tier as everything up in the obsessed category, so it's going down into the interested category. And finally, we have the final protein. Oh boy, I'm out of breath. I don't know how streamers do super long streams. It's so tiring. And that is scramblase. So where... when I ported these images into this particular Photoshop picture, I don't think all of them got sent over, which is a little iffy. Okay, you know what? If I'm gonna make exceptions for ribosomal proteins and scramblase, I'll go back to do bilirubin too, just so that we do as many of the viewer proteins as possible. So we'll do scramblase. Okay, so we'll do scramblase. Oh, wrong layer, scramblase. Okay. So scramblase has a similar appeal to me as the snare proteins. So scramblase also have to overcome the problem of, if you want to move lipids, so instead of vesicles, we're talking about lipids. If we're talking about lipids, you still encounter that same issue, right? Same issue. Actually, I'll just blow up the snare protein image again because it was so helpful. You still have this issue where if you, instead of vesicles, you just want to move like fats across the membrane. If you just want to move fats, that's still difficult because of the same reason, right? Positive repels positive. That's not like energetically favorable, so the cell has to create clever little solutions to get around that energetic barrier. And one of those ways is with this enzyme called scramblase. And yeah, more juice. I think even Riley had said when they had suggested this protein, the name alone is just hilarious, right? I also put in my notes, good name. So scramblase has the ability to overcome the massive energy barrier that trafficking lipid demands. And a part of this class of enzymes are things like flipases and flopases, which are also great names. And so basically, I don't know enough about their mechanics to actually explain how they work, but they solve the same problem that snare proteins do. So for that reason, I'll put them down, and I just don't know enough. What Riley did mention is that they are involved in cardiolipan biosynthesis, which has a role in apoptosis, which is kind of interesting, but I don't know enough about this yet. I'll re-look at these proteins and probably re-tier them at the next tier list. I want a hotel breakfast right now. I'm leaving. Yeah. Scrambled egg ace. Yeah. Oh man. If you haven't like watched my weird gene name short, you definitely should. Like, biologists have a lot of fun naming things. So I'll go back to talk about bilirubin, which technically isn't a protein, I think. I think it's a small molecule of some kind, but we'll do it anyway. Bilirubin. Right, bilirubin. Just make sure I spell that right. So this is a breakdown product of heme, and it might have antioxidant function. I really struggled because I didn't know what B wanted me to say, but what I gathered was that this is a molecule that is responsible for the yellow coloring of healing bruises, which I thought was kind of neat. So yeah, bilirubin is going to be put into the don't know enough category. I will re-tier these the next time I do one of these tier lists. And finally, we have made it to the end of the first all proteins tier list, which I'm very, very, very excited about. I hope you all had fun. I had a lot of fun. I didn't get to quite talk about every individual protein in as much depth as I wanted to, but that's because, you know, one more acknowledgement that biology is very complicated. There's a lot to learn, a lot of stories. And to come prepared to talk about 30 different stories is like bonkers. But I hope this was pretty good in terms of giving you a little teaser into the exciting stories that I want to elaborate on in the future. Bilirubin, is that at all related to bile? I don't know. It could be. All these proteins look delicious. Get your macros, right? I'm gonna take a look at this poll, and yeah, as I thought, mitochondria wins favorite organelle easy. The reason why I put up this poll is because I want to attempt, no promises, bi-monthly streams where I cover the AP biology textbook for high school, but I take one interesting story and really dive into it. So this was kind of a way for me to suss out what organelle I should talk next time I stream. But thank you all for coming. Thank you for participating. I hope you found everything to be interesting. And one more plug before you all leave. I have another stream. I'm probably not going to do 2 streams in a week ever again because of how much work these streams are. But I have another stream coming up, and it's with a fellow educational VTuber called Sidere. who's a friend of the channel, and that will be happening on Sunday at 10 a.m. EST. The thumbnail is already out, so it should be on your subscription pages already. She's an awesome, very fun, very different style of educational VTubing than I do, and I hope you check her out. I don't think I put her link in the description because I'm bad, but I will. Did I? I think I did. No, I didn't. Okay, I'll plug her right now. You can find her here, and she does like similar, a kind of a similar idea, which she'll talk about very high level science, but in a way that I think is very accessible and fun. And she has just like a different energy than me, which is very fun to see. With that, I think I'm going to call it a night. Thank you all for coming. I hope to see you on my collab stream. Oh. One more thing that I want to mention is that I'm not going to end up actually releasing my Plague Inc. short tomorrow, just because A, this is the last Plague Inc. short that I think I'll make for a very, very, very long time, maybe indefinitely. But that's because I want to get the science 100% correct, is that I think in my original script, I may have been propagating misinformation and maybe potentially offending some people, and so I just want to make sure that I don't do that. So if you were looking forward to the end of the Plague Inc. series, I'm sorry, that's going to have to wait for a while. But yeah, thank you all. I had a lot of fun, and thank you for 1K cellmates. Let's go. 1K is such a huge milestone, but it does mean that I will have to eat a SCOBY at some point in the future. Thank you all for watching, and I will see you next time. Bye!